Thank you, Bruce, and thank you to the entire society and the committee that nominated for me for this award. It's really an honor to speak as a part of this symposium and also to be acknowledged by the award in the name of Britton Chance. Britton has probably affected everybody's life in this room, and we worked on a number of things that Britton was interested in and would have me out to UPenn to have conversations about in vivo bioluminescence imaging and imaging photons. And it's really an honor to be in the shadow of such a giant in biomedical optics. So in vivo bioluminescence was really started because we wanted to enhance discovery. I call it the tail of discovery because most of what we inject into mice goes through the tail vein. And the biological source of light that we use is this enzyme, or one like it. It's an enzyme requiring oxygen and can be used to sense in vivo or to be used as a reporter gene. The DNA from this organism and organisms like it can be placed into animals as a marker of biological function. And by labeling these gene pathways, we can look at gene networks, we can look at expression patterns, we can look at biology in real time in living animals. I got into imaging because I wanted to track viruses. So we would label viruses with luciferase as well and track them. But it's quite a versatile technology. Bruce mentioned that my wife Pam and I worked on this together. She was interested particularly in bacteria, and those can be labeled with a different luciferase, one derived from a bacterial organism. And the majority of the work done in this field is really on mammalian cells, labeling tumor cells or stem cells and watching them traffic in living animals. The simplicity of this technique is really what gave it its leg in the field, simply injecting the substrate into this animal and placing it in a box where we exclude ambient light allows us to image biological change, in this case, the growth of tumors in a living animal. There were three of us that were involved in the early days, and Bruce mentioned David Benarin and my wife Pam. David was just really an incredible colleague in putting this initial project together, and Pam was, of course, interested in bacterial pathogenesis, and the three of us together came up with these tools and technologies. David had the privilege of studying with Britton during his career, and I think when Britt and I had conversations about bioluminescence, he would give me the looks like you see on the far left and the far right of really thoughtful consideration. And every time you mentioned David Benarin's name, he'd give you that smile, that affectionate smile that you see on the second image to the right. I think it was because of David's brilliance that Britt would give that affectionate smile. So David and I assembled the first in vivo imaging system to do this back in the early 90s. And the three of us founded a company, XenaJen. And the first thing we did was recruit two really incredible engineers, Mike Cable and Brad Rice, and they made the system what it looks like today. The system that David and I built was really a sheet metal box with a camera stand from Keeble and Schuckett Photography, a very simple system. But really, it's its versatility and simplicity, I think, that really attracted Britt to thinking about this problem with us. I've been really grateful for the people I've worked with at Stanford for the last 20-some years, and the team at XenaJen, which grew into a fairly substantial development team, and as Bruce said, transitioned to Caliper and now is part of PerkinElmer. Just by way of disclosure, I don't have any stock in any of these companies anymore, so this isn't a shameless advertisement. But the reason that this technology was so powerful is that it really gave us new information that we didn't have before. And I like to show this movie because it emphasizes all the things in blue that imaging helps us do. But I like to show this movie for another reason, and that is these are composite images taken every 2 hours for 2 days, and then every 4 hours for another 4 days. And they were taken by a single undergraduate who lived in the lab for a week taking these images. But now he's a practicing physician treating patients, even though he had his roots back at Stanford in bioluminescence imaging. When we started this study, I remember telling somebody, or telling people that I wanted to study gene expression patterns in living animals and watch them in real time, and people said that's not possible, you can't do it. And now undergraduates, starting with Chris Gruber back in the late 90s, who generated these images, within a week, undergraduates are watching gene expression patterns in living animals. This gives us access to new information because now we can look at the changes in the context of the host where they're intact. We get more information per animal. We can do whole body scans. So we can ask where things are changing and look at different organs. And we can use that information to guide us to the right time and the right tissue to sample. Statistics are improved, and image guidance allows us to now guide our selection of tissues for omics. And since the reporters can be used both in culture and in vivo, you can go between cell culture and animal models and back and forth to improve the studies. So because of the simplicity of the system, it really opened a lot of windows into mammalian biology. And I'd like to show you just a few examples that emphasize some of the things that have been discovered because of this ability. This is a model we set up a number of years ago with my colleague at Stanford, Rob Negrin. And it's a lymphoma model where we're just asking, what happens to lymphoma when you treat it with radiation and give the animal a bone marrow transplant? This is exactly the way a child with lymphoma would be treated. So we initiate the cancer with 3,000 cells, image at 7 days, whole body lethal irradiation to kill the tumor, and because it also kills the bone marrow, you give a bone marrow transplant. Image again, you're seeing what happens with this therapy. You see a reduction in the primary lesion. This is all what you would expect from this model, but it's always the unexpected result that's the most interesting. And what we observed if we followed these animals over time is that we could treat the disease in the periphery, but because of the bone density, the lymphoma would persist along the spinal column in these animals. So we looked at this and said, well, this is a real opportunity for us now because we can begin to look at the cells that compose minimal residual disease, the cells that are resistant to conventional therapies. In addition, we can pick the bone marrow transplant therapies apart. Rather than transfer whole bone marrow, we should just transfer the stem cells. Those are the cells you need for reconstitution. We should just transfer the memory T cells, which are the ones that would protect the animal or the child with lymphoma from infectious agents. In addition, there are cells in our body that are tumoricidal, that kill tumors inherently. Natural killer T cells and natural killer cells. So if we could just refine our bone marrow transplants to these three populations, we'd have exactly what we need. And so we've looked at all three of these populations in some detail. We initially started looking at just whole bone marrow transplant. And so this is the first time we've been able to watch bone marrow transplantation in real time in an animal model. I love this image because the images are really striking, but it doesn't tell us too much about the biology of the stem cell. I'm really interested in what happens in the time point between that first image at 1 day and the middle 4 images at 4 days. What happens in that time point? What happens to each individual cell? Because again, this is the expected result. You expect the bone marrow cells to go to the bone marrow. You expect them to go to the spleen. Those are the sites of hematopoiesis. So we started using fewer and fewer cells. Rather than transferring 5 million whole bone marrow cells, we dropped the numbers down to 250 stem cells, 50 stem cells, or 10 stem cells. And the images on the top are from those really small numbers of cells, 10 stem cells. We have to transfer unlabeled cold bone marrow at the same time, otherwise the animal would die of hematopoietic failure. But what you see on the top are the initial foci. You're looking at the foci, the groups of cells that start from individual cells. And so we can begin to ask, what is the frequency of engraftment? Where do they go? And how well do they perform at different anatomic sites? In the bottom panel, you see on day 6 that there were 2 cells that engrafted in this animal. And if you put 1 stem cell in each of 100 animals, you get about a 20% engraftment efficiency. And here, when you put 10 cells into a mouse, you see 2 foci, again, 20% engraftment efficiency, and we saw that with all these numbers. But what you'll notice in that animal on the bottom, which is followed over time, is that one of those foci participates in hematopoiesis. You see it grafting in the skull and then reconstituting the entire animal. The other focus seems not to do that. It moves. It doesn't stay where it was at day 9. You see it in a different place, and again it moves again. So even though we started with very small numbers of cells, we couldn't really pick apart the entire pathway. So after the study, we decided to use a single stem cell. And so what you're watching here is hematopoiesis driving all of the blood cells from a single hematopoietic stem cell. On the top, you see there's a cell that successfully engrafts and induces hematopoiesis across the entire body. The stem cell on the bottom does not. And now we can begin to understand what those pressures are on the cells that allow them to participate or not. And using that, we've identified a number of genes that play a role in a stem cell's decision. When it divides, does it become another stem cell, or does it differentiate into another cell type? And so we use this approach to identify a number of genes that play a role in that process. The roots of this technology are in imaging of infection. And every time we set up a new model, we learn something new about a disease that we've been studying for tens, or even in this case, 40 or 50 years. In this model, this is an infection with listeria. Listeria was thought to be an obligate intracellular pathogen that only caused an acute infection. But what we're seeing here in this image is that it caused an infection in the gallbladder that persisted for days, weeks, and sometimes months. And what you're seeing in that little window is the extracellular bacteria growing outside the cells. So for the first time, we're able to observe that this bacterium can grow outside the cell in the animal host. In addition, we could find it in other anatomic sites because of the sensitivity of detecting these labeled pathogens due to this signal to noise being significant in this approach. We found that it also caused osteomyelitis. You could find the bugs growing in the bone marrow. If you remove the bone marrow and section it, even though they also express GFP, we couldn't find it by fluorescence because with these images, we're estimating there being about 200 bacteria at that anatomic site. So the sensitivity of detection allows us to find subclinical infections. The reason this was important for this particular pathogen is that this is a pathogen that causes abortion and preterm delivery in humans. It also causes meningitis. Until we observed the persistence in the gallbladder, people thought that the food contamination from listeria would come from the environment, but it's likely the case that food handlers are infected with the pathogen, and it's infected their gallbladder. They go out for lunch and get a big cheeseburger and have all that fatty food, then induce cholecystokinin. The gallbladder contracts, and you can actually, in these models, inject cholecystokinin and see the bacteria track through the intestine. So after the gallbladder contracts, it fills up again, and the listeria are still there. So you have this constant release of bacteria into the intestine. This is probably a subclinical infection in most cases and doesn't have any tremendous impact on the person who's infected, and we've now identified that this does happen in humans. But it is a source of food contamination. But even more importantly, listeria is a very important pathogen for pregnant women. It causes, like I said, preterm birth and a variety of developmental delays. So Jonathan Hardy in the lab set up a model for looking at the infection of placentas by listeria. In the placenta, it does grow intracellularly, and you'll see in that beautiful confocal image from Anna at UCSF that you can see the listeria in the placenta, and in the image on the far right, very lower corner, you see the bacteria growing intracellularly inside a macrophage. In this animal that you see, the pregnant animal you see on the far left, there's no signs of maternal infection. For the mother, it's a subclinical infection. This probably happens all the time in the human population. But I guess those movies aren't going to play. So let's see if I can get those to go. So what we observed in these animals, if we did ultrasound on the animals, is that the low level of infection would cause a bradycardia, would cause a slowing of the heart rate, and you can't appreciate those in the ultrasound images because the movies aren't playing. But we can compare in this animal the animals with an infected placenta to those that are uninfected. In this particular case, only one uterine horn had infected placenta. So we also observed that we could mimic what happens in humans. We could get preterm delivery, we could get craniofacial deficits, all the birth defects that neonatologists worry about as a result of infection can be modeled by this pathogen, and we could begin to understand the mechanisms by which this occurs by looking at the infected placentas right next to an uninfected placenta. So this is a versatile and simple technology that was disruptive in the field. It fundamentally changed the way that animals are studied. It refined the model so we could produce more and better data and do it faster using even fewer animals than require using conventional approaches. And for that reason, it's been adopted widely in the drug development across a variety of different areas. This is a slide put together by the group at PerkinElmer demonstrating the number of companies that have used the in vivo bioluminescence imaging to advance drug development. This is a sample of some of the drugs that have actually gone through FDA review using in vivo bioluminescent data to support the FDA application. So a very simple idea that began some 20 years ago now is having an impact on the drugs that are being used to treat cancers and a variety of other diseases. So this is one tool in a toolbox of imaging technologies that's had a dramatic impact on how we study mammalian biology. Here you can see that ultrasound image playing, showing a normal heartbeat in a fetal mouse. These tools are used in combination with single photon computed tomography and CT, cardiac gated CTs, PET imaging with CT, and one of the more current optical imaging tools you see on the lower right, where it's combining CT with optics so you can begin to localize the signals in three-dimensional space and not just in 2D overlays. So this has really changed preclinical drug development across a variety of different indications. So that was the background that led us to think about developing tools that we could actually translate into the clinic. I remember when I would give this talk on bioluminescence imaging 15, 18 years ago, people would ask, when is this going to be used in humans? And I was always scratching my head wondering how I could use this in humans and couldn't come up with a really great way to do it. It's probably possible, but there are tools that we can translate to the clinic and use what we learned from animal models to think about how to attack a disease in humans. So some of the tools that we're developing in my group for clinical translation include miniature confocal microscope. This is an effort led by Mike Mandela in my group. The idea of optically sectioning the tissue through an endoscope. And the instrument you see here is our smallest confocal microscope. The way these devices work is that we use MIMS mirrors that have two faces to scan illumination and a collection beam into the tissue. And we can run just one scan mirror and do a needlepoint scan, poking through the tissue looking for pathology. If we find something of interest, we could run the XY scan mirror and optically section the tissue, and again, do this in living patients. And if we had the time to really pick apart the biology in that particular environment, we could run both the Z-scan and the XY scan mirrors and create a three-dimensional image of the cells and the relationship of the cells in that tissue where we find the unique pathology. So here you see a version of that, a larger version of that microscope being used in the clinic. This is an effort led by Shai Friedland of Stanford. Tom Wang and Zhen Kui were at Stanford at the time, and now they both were at Michigan, and then Zhen has come back to work with me at Stanford. And Heijun and Weibel have both contributed by designing these mirrors, but all of these people were in the endoscopy suite as we were testing these tools, asking what more can we do to improve them. So you're seeing here the endoscope image on the right and the microscopic imaging on the left so that we can combine the two and guide the selection of the tissue for pathology and guide the biopsy for the right tissue for sampling. Most of what we've done in the lab has been with bioluminescence and fluorescence, but we've been a little bit frustrated with the ability to multiplex those approaches. And so we've begun to both make the microscopes faster and cheaper. Frank Schoenig came to the lab and he said, why are you micromachining these microscopes and assembling them by hand? Why don't you just print them on a wafer? And so he asked us to see if to look at the possibility of using a lithographic approach or a liga. Liga is used to make watch parts, and so we thought that'd be perfect for making microscope parts. And here you see all the parts that went into about 5 microscopes. Those parts come off in these as these little pieces that can then be snapped together, much like a model car that we built as kids. I put it in a little jewelry box here because it's more valuable than any jewelry I have in my house. But those little pieces snap together, as you see in the figure D, and then are covered with a gold housing. This is an instrument we developed for as an in vivo implantable confocal microscope, where we can put the microscope right on the tumor and leave it implanted in the animal and follow this animal over time. So as I mentioned, we were frustrated with the inability to multiplex with fluorescence and bioluminescence. And so we started developing miniaturized Raman endoscopes. This is one that we designed to scan the human colon in 10 minutes or less and try to multiplex with a variety of different surface-enhanced Raman particles. And Ellis Gray, who led this effort with John Liu's help, was a very thorough investigator. So you see his panel of all the different combinations of Sears particles mixed in different ratios, different combinations, and imaged with this scope that can be used in the clinic. And you see him imaging here in the human colon. And so we've begun to develop a suite of technologies that allows us to go from mouse to man and begin to ask some of the questions that we could ask in our animal models in humans and develop the tools to advance that field. There's no way I can thank everybody that's contributed to the work I've talked about today. Here's a few of the people that are currently in the lab, some previous lab members whose work I talked about, and our many collaborators that have contributed to this effort. And with that, I'd like to thank you all again for your attention and for this opportunity to present this work on behalf of my colleagues.